Assalamu alaikum. I'm Hasina Uzair along with Javeria Wadood, and today we are going to discuss about the organic compound analysis by the various titration techniques. These are the contents which we are going to discuss in the coming slide. The first one that we are going to discuss that how dose titration can be helpful in determining organic compound. Secondly, also the application of the acid base and the complexometric titration in the analysis of the organic compound. So, Javier is going to help you in understanding the various methods of how organic compound can be analyzed using the dose titration.
In this slide, I'm going to discuss about the redox titration before getting into the redox titration application. So basically, redox titration is a type of titration that is based on the redox reaction between the analyte and titrate. In this type of titration, reducing agent and oxidizing agent are involved, and that helps in analyzing the analyte. In our next slide, we are going to discuss about the application that are based on the redox reaction. Walkie and black method is a titrometric procedure. In this, soil is first oxidized with the excess of dichromate, which is the oxidizing agent. Along with that, sulfuric acid is also added. And from the residual dichromate that left behind while titration, is titrated back with the iron sulfate, and in this way we can calculate the concentration of the organic compound in the soil. Here we have the procedure for the Walkie and black method. First of all, 500 ml of the flask is filled with soil, into which 0.167 molar potassium dichromate is added. Along with that, H2SO4 is also added and it's stirred until uniform mixture is ensured. Here is a chemical equation for the reaction. Potassium dichromate that acts as an oxidizing agent is converted into chromium ion, with which carbon dioxide is also released and water molecules are produced. Not all the potassium dichromate get reduced into the chromium ion. The rest that is left unreacted is get reduced into chromium ion by titrating it with the ferrous thiosulfate. And in this way, from the concentration of chromium ion, we can calculate the organic analysis within the soil. Determination of ethanol in aqueous solution. So here is a procedure for the determination of ethanol in aqueous solution. Ethanol can be determined in the aqueous solution. Aqueous solution can be the sample of beer or any alcohol. So first of all, excess of potassium dichromate is used to oxidize ethanol into ethanoic acid. The amount of unreacted dichromate is then determined by adding potassium iodide solution, which is also oxidized by the potassium dichromate forming iodine. The iodine is then titrated with a standard solution of sodium thiosulfate, and the titration results are used to calculate the ethanol content of the original solution. Here we can see the two beakers that containing two solution different colors. So this is due to the iodine that fades the solution from dark brown to yellow. When the brown iodine color fades to yellow, starch solution is added and keeps titrating until blue color disappears. And in this way, we can calculate the concentration of ethanol in the aqueous solution. In next slide, Hasina Uzair is
I'm going to discuss acid-base titration and how it can be useful in the determination of the organic compound. Basically, it is a type of titration in which a reaction between the titrant and analyte involves acid-base reaction. For example, an acid may be a solution with known concentration and is reacted with a base of unknown concentration, and in this way, the unknown concentration of base can be determined. So we can take a base with known concentration reacted with an acid of unknown concentration, and in this way we can determine the concentration of an acid. Now, how can we determine the amino acid and protein in an organic compound? For this, I am going to discuss the two methods for the determination of amino acid and protein. The first one is the Jedhal analysis. It provides a method for the protein analysis. In this method, nitrogen is estimated by the ammonia gas. The organic compound is heated with sulfuric acid. Nitrogen in the organic compound is then converted into ammonium sulfate as shown by the fumes of the gas head. The ammonium sulfate is then heated with excess NaOH and it liberates ammonia. Here you can see the heating of the solution and the liberation of the ammonia gas, the ammonia. Here the fumes of gases is the ammonia gas, and the ammonia gas is dissolved in standard solution. Sulfuric acid is taken here. The produced ammonia can be estimated by the amount of sulfuric acid used in the reaction. It can be done by estimating the amount of sulfuric acid left after the absorption of ammonia by titrating it with standard alkali solution. The difference between the initial amount of sulfuric acid taken and the amount that left after the reaction provides the amount of acid reacted with ammonia. Now, in order to find the percentage of nitrogen present in ammonia, we use the following equation. And this equation 1.4 shows the molar mass of the mass of the nitrogen, while m is the molarity of the H2SO4, and V shows the volume of H2SO4 taken in milliliter, while V1 is the volume of the NaOH, which is also taken in millimeter and is divided by the mass of the organic compound. The second one method is the determination of the primary amine. The primary amine is determined the pentene dione method. In this method, excess of pentene dione is taken and is reacted with the primary amine in the pyridinene medium. It gives us the amine along with the unreacted pentene dione as it was taken in excess. Then the unreacted pentene dione is titrated with standard solution of the sodium methoxide using the indicator. From this, we can determine the concentration of the primary amine, and we can determine the primary amine by this method. Here, this shows the reaction in which the 2,4-pentene diol is reacted with the primary amine, giving us the amines. Here, the pentene dione is a weak acid, while the amine, which are the products, are the essentially neutral. Therefore, in this method, the pentene diol is taken in excess and is reacted with primary amine, and its concentration is determined. This is the indicator which was used in this method, and the formation of the blue colors represents the end point of the reaction. Then secondly, how can we determine the functional group? Most commonly, iodometric titrations are used for the determination of the functional group. The carbo has a certain weak acid containing the functional group of carboxylic, sulfonic, and phenolic are usually treated in either aqueous or non-aqueous solvent, while the aliphatic and aromatic amines are weak bases, so they can be titrated using HCl in aqueous solution or HCl4 in industrial acetic acid. While the other functional groups can be analyzed indirectly by the use of the functional group reaction that produces or consumes an acid or a base. As an example of how we can determine the carboxyl functional group, the procedure is here as follows. You have to place a known amount of purified compound in a conical flask. Then you have to dissolve it in some ethanol and dilute it with deionized water so that the precipitation does not occur. Then you have to add a few drops of the indicator and titrate it with standard solution of the NaOH until a faint color appears. Faint color basically represent the end point of the reaction. Then you have to note the readings which was used until the pink color appears. You have the this data. You have the weight of the compound. You also have the known. You also know the volume of the NaOH taken and the molarity of the NaOH as well as molecular weight of the compound. By computing the values in the equation, you can determine the number of the COH groups. Here the V represents the volume of the NaOH taken as a titrant, while M is the molarity of the NaOH, and this is the molecular weight of the compound divided by 1000 along with the weight of the compound. Now, the third one application of acid-base titration is that it is helpful in the determination of the volatile fatty acid. Basically, volatile fatty acid is a group of different acids, including acetic acid, carbonic acid, butyric acid, and valeric acid, being the most common form. The dominant among them, the dominant volatile fatty acid species in wastewater is acetic acid. The volatile fatty acid plays an important role in the denitrification. Also, and it has an enhanced role in the biological phosphorus removal, and they can be used effectively for the control of mesophilic sludge digesters. The basic principle for this method is that the acid required to titrate a sample from pH 5 to pH 4 can be considered proportional to the content of the volatile fatty acid present in the sample. The procedure for this method is that before analysis, you have to filter the sample using the 0.45 micromembrane filter. Then you have to take 20 ml out of it and put it in a titration vessel and record the initial pH. Then you have to slowly titrate it with the sulfuric acid, and you have to record the added volume until the pH reaches to 5. Then you have to slowly add the sulfuric acid until the pH reaches to 4.3. The total volume of the added titrant is again recorded. And the later step is repeated. This step is repeated again and again until the pH 4 is reached and the volume of the added titrant is recorded once more. In this way, we can determine the volatile fatty acid content present in the sample. Now, Javiria is going to discuss about the complexometric titration application in the determination of the organic compound.
In this slide, I'm going to discuss about the complexometric titration. Complexometric titration is a form of volumetric analysis in which the formation of the colored complex is used to indicate the endpoint of the titration. Complexometric titration has various applications and therefore is used in determining the carboxylic group in cellulose. The ion exchange properties of the cellulose fibers are mainly determined by the content of the carboxylic group, and these properties can therefore be used to determine the number of accessible groups, which is a matter of special importance when determining, for example, oxidation processes and the polymerase reactivity. In this way, carboxylic group is determined in cellulose. In the next slide, we have the procedure for determination of the cellulose fibers. So first of all, cellulose are first oxidized. Cellulose fibers are oxidized either through selective oxidations or through non-selective oxidation process. Similarly, the samples that are oxidized are dried and is weighed, into which 100 ml of calcium acetate solution is added and is shaken and kept overnight. The solution that has been prepared is titrated with EDTA solution, while the pH of the solution is adjusted by sodium hydroxide that is added about 0.1 molar. The solution that has been made is prepared into beaker along with the indicator urate site, and the suspension is then filtered. The equivalence point can be determined photometrically using Mettler-Toledo DPC60 photoelectrodes, which measure the absorbance of the solutions after each addition of the titrate volume. Hence, the color changes in the equivalent point can be perceived automatically. This was all from our side. Thank you so much.